brain. So these are very important. But before that, the antigen presenting cells, we are also called as a macrophage, or we are also called as a phagocytic cells. These cells are analyze the antigen, or recognize the antigen and analyze the antigen, and engulf the antigen and separate the two substrates of the antigen. One we are called as a non-toxic material, and the second one we are called as a toxic material. So non-toxic material is dissolved in our blood, but toxic material is not dissolved in our blood. It is remaining contain on MHC2 molecules of the antigen presenting cells, which are present on membrane of the APC cells, or antigen presenting cells. That MHC2, the MHC2 contain this toxic material and present to the CD4 plus of the TH cells. So when the TH cells gain the signals, or attached or bind the antigens with the help of CD4 plus, then the TH cell is activated. TH cell is activated and release some molecules, that we are called as a interleukin 2 molecules, or we are also called as a cytokines, that we are called cytokines. So these cytokines are intermolecules. Interleukin 2s are very important for the activation the B cells. They activate the B cells. Then the B cells release the antibodies. These antibodies fight with the antigens and neutralize the antigens. So this is the activation of the TH cell with the help of antigen presenting cells, and activate the B cells to release the antibodies. And also TH cells also act as a presenting the antigens to macrophages. So sometimes TH cells also present the antigens to macrophages. This is reverse process. And again they are act as a killer T cells. Sometimes they are also kill the antigens. Kill the antigens and neutralize the antigens also. So they are very important in the immunity to activate the TH cells and cause to release the antibodies. Hello students, in this video the topic is B cell activation. So the B cell activation is very important for the binding with the antigen and neutralize the antigens. So these B cells are released from the bone marrow. So this bone marrow release the B cells after the maturation is complete. Actually bone marrow is very important for the produce the B cells. Then the B cells are produced and mature and released into blood for the immunity. So this is the very important about the B cells are released from the bone marrow. So when the B cells are released and ready for fight with antigens, when the antigens are released or antigens are enter into our body, the B cells are normally bind with antigens with the help of B cell receptors. So this is normal process by the B cells. So then, but in sometimes the antigens are presented by the another cells that we are called as a macrophages, or we are also called as a phagocytic cells, or we are also called as a antigen presenting cells. So these macrophages or antigen presenting cells are very important for the present the antigens to B cells. So it is a step by method is step by step method is present in the presenting the antigens by the macrophages. So the first one is by the macrophage. Suppose macrophage is there, Actually, first one is it is recognize the antigen. It is recognize the antigen. That is first point. And the second one is engulf the antigens. Means it is that is cytosis. Means phagocytosis the antigens. Means gain the antigen into the cytoplasm of the macrophage. After gain the antigens, that is destroy the antigen. That is the third point of the macrophage. Destroy the antigens means the antigen is destroyed and separate the two substrates, that is called as a non-toxic material and called as a toxic material. So this toxic material is again present on the membrane of the that is macrophage, but the non-toxic material is neutralized and soluble in our bloodstream. So because it is non-toxic material, it is not harmful to our immunity or our body. So the toxic material is should present on the membrane of the macrophages, on the macrophage. So this is macrophage and it is present on the macrophage membrane. So this antigen is present to the B cell receptors of the B cell. So this is the antigen present. So this is the fourth point of the macrophages. After the that is presented by the macrophages, B cell is activated. That is fifth point. That is fifth one. So when the gain the B cell naturally or by the macrophages, the B cell is activated. That we are called activated B cells. So this is now activated B cells. So after the activated B cells, it is ready to that is clonal expansion. That we are called as a clonal expansion. So means produce the different type of cells. That we are called proliferation. So that process we are called as a proliferation. So when the activated B cell is there, that is activated and produce the different type of cells, that we are called plasma cells, and another we are called memory cells. And also B cells activated by another cells, not by their own and not by macrophages, and not by own, but it is also activated by T helper cells. This is called T helper cells. So T helper cells also that is gain the antigens sometimes with the help of macrophages or by the own of the T helper cells, and engulf the that is antigens and destroy some that is non-toxic material, and toxic material is present on the CD4 molecules of the T helper cells, and it is present to MHC2 of the B cells. So the antigen is present by the CD4 to MHC2 B cells. So by this process the B cells also activated. After activation is completed, the clonal expansion is there and proliferation is present. So after proliferation, the two types of cells are produced. One we are called memory cells and second one we are called plasma cells and memory cells. So the plasma cells we are called as a first line of immunity or first primary immunity. Why? Because these plasma cells are fight with antigens by the release of antibodies. So means within short time the plasma cells release the number of antibodies, and these antibodies bind with antigens and destroy the antigen within 10 minutes or 20 minutes or that is in the 6 hours or 9 hours, means that is before the 20 or within 24 hours it is destroy the total antigens. But along with the plasma B cells, the memory cells also proliferated. But if these memory cells are not release the antibodies, but they remain the antibodies in their surface and wait for the next entry of the again entry of the antigens. So this we are called as a secondary immunity. Secondary immunity. So means that is suppose for example malaria is enter into our body, then the B cell is activated by the macrophages or T helper cells or thereby own their cells, and after that the B cell is activated and release the plasma cell. These plasma cells are destroy the Malarian antigens. So the person, that person is cured by the malaria. So is the immunity is gained by the plasma cells and the antigens are destroyed. He is escaped from malaria or he is covered by the malaria. But after 1 month or after 2 months or after 1 year, again the malaria is attacked to same person. But this time the B cells are not activated their own, because already memory cells are present in our body. The B cells are not activated and they are not again enter into proliferation, because the memory cells are already present and they are directly attack on antigens, because release the antibodies, and they are directly attack on antigens and destroy the again same type of antigens. So means malaria. So this is called as a there's a secondary immunity, that is called as a secondary immunity, but it is a by the plasma cells. Primary immunity, this is very important. The primary immunity is the first entry of the antigen. Secondary immunity is by the second entry of the antigens, that is by the memory cells. So this is the B cell activation. The B cell activation also called as a humoral activation. The B cell cells activation also called as a humoral mediated cell activation, because the B cells are not directly kill the antigens. They release the antibodies. That antibodies kill the antigens in our bloodstream. So that's why the indirectly antibody kill the antigens in the blood. That's why we are called as a humoral mediated antibody. Humoral mediated, that is immunity or antibodies, immunity or activation. Right? So this is the B cell activation. Dear students, in this video about the types of antibodies. So here after the structure of the antibody or immunoglobulin, the types are also present in the antibodies, so that we are called as a gamma. So as a simply we are called as a gamma. So these 5 types of immunoglobulins are present. So one we are called G for gamma and A for epsilon, M for mu, D for delta and E for epsilon. So alpha, gamma, mu, delta and epsilon. So this is the types of antibodies. Now you see in the image. So these are the structure of the antibodies. Easily identified that this IgG, so immunoglobulin G, that called antibody G, antibody E, antibody D, and this is antibody A and antibody M. So immunoglobulin alpha. So actually immunoglobulin alpha has 2 antibodies. They are joined with J chain. So that is called a joining chain or a chain. The joining chain is present between the 2 antibodies, and at the same time another component also bind that 2 antibodies in the antibody A, that is IgA, that is secretory protein. So means every that is between the 2 antibodies or between the 5 antibodies in the antibody M, that is immunoglobulin M, there are 5 antibodies are present. So this is 4 and 5. These 5 antibodies also connected by the that is a joining chain. that is connected by the joining chain, or that is disulfide bonds that we are called secretory proteins. So this middle part, they are connected as a joining chain connected by and disulfide bonds. So these are the 5 types of antibodies. Actually, the IgM we are also called as a pentamer because 5 antibodies are present in the IgM. It is a big antibody, and IgA there are 2 antibodies are together by the joining chain and secretory protein that we are called as a dimer. So this is called dimer, and this one we are called pentamer. So this is called pentamer. So but IgG, IgE and IgD, so these are single structures are present, so these are called as a monomers. So this we are called as a monomer. So monomers. So these are monomers, these are pentamer and these are dimer. So these are the types of antibodies, and we are observed that every character of the antibody. So in the next image, that is this is the different type of that is characters are present in every antibody. That is we are called this is IgG, that is monomer, IgM, this is pentamer, IgA dimer, and IgD and IgE, that we are also called as a monomer. So that is types of antibody. So how they are differentiate the types of antibodies means they contain different number of amino acids in their heavy chains. Suppose these blue colors we are called as a heavy chain. So in this and in this, the blue color heavy chain, in the structure of the antibody, heavy chain contain so different number of the antibody, that is amino acids. So that's why they give a name based on the amino acids, based on the heavy chain. They called that is gamma, mu, alpha, delta and epsilon. So based on this, they give as a IgG, IgM, IgA and IgD and IgE. So these are the 5 types of antibodies, and that is number of antigens are bind with each one antibody. Suppose already we are discussed in the structure of antibody, that is in the structure of antibody the number of antigen. Suppose in the IgG, suppose observe in the IgG there are only 2 antibodies are bind like this, so that's why they accept only 2 are bind with 2 antigens. But in the pentamer of the IgM there are 5 antibodies are there, each antibody bind with 2 antigens, so 10 antigens are bind with that is IgM. And in the IgA there are 2 and here 2 antibodies are bind with that is IgA, so totally 4 antigens are bind with antibody. And in the IgD and IgE 2 and 2 antigens are bind with antibodies. So highly antigens are bind with IgM and IgA. So these are the second feature of the antibodies. So differentiations, and the next differentiation is that there is a molecular weight of the antibodies. So in the molecular weight of the antibodies, the IgG is a 1,50,000 molecular weight in deltas, and IgM 9 lakhs. Why? Because it is a very big size of the IgM is there, that is pentamer is there, that's why their molecular weight is 9 lakh is present. And IgA, so IgA is 3,85,000 and IgD 1,80,000 and IgE 2 lakhs molecular weight is present in the molecular weight that we are called deltons. So these are the third type of antibody character. And next one, that is we are called present of the antibodies in the plasma serum. So that is IgG is present as a 80% in plasma serum. and IgM is only 6%. They are only 6%, but they are in structurally very big. And there's IgA. So IgA is only 13%. IgD and IgE below 1%, they are present in our total plasma serum. So these are the that is fourth character of the antibodies. And next one is that is the function of the antibodies. So main function, that is IgG is cross the placenta. So this IG antibody is IgG antibody is crossed from the mother to children's. So that's why this only the IgG is only cross placenta, but remaining four is not cross the placenta. Between they are not transfer between mother and children or that embryo. And so in the complement system, that is complement system means when the antigens are enter into our body, our some components are released to destroy the antigens, that we are called as a opsonization and agglutination. So in the opsonization means that we are called destroy the antigen process as a process, the step by step process. In that step by step process, there are some molecules are released. That molecules we are called as a complement molecules. The type of complement molecules are released in the IgG and IgM only, but remaining three is not released. That complement molecules are fixed complements, that we are called fixed complement. So these are the sixth or that is another type of function of the antibody. And the last one, the last function of the antibodies is that every antibody is their own function is there. So IgG is there. So IgG is a function as a neutralization. The antigens are neutralized by the IgG antibodies, and also agglutination. They form a clump-like structures in the along with the antigens. This type of antibodies, and complement activation is also present. That already we have discussed in the fixed complement, and opsonization, so cell death. And this IgG is depend on cell mediated toxicity. So it is depend on cell mediated toxicity. These are the main function of the IgG: neutralization, agglutination, complementation, and opsonization, and antibody dependent cell mediated toxicity. So this is the IgG functions. These are the IgG main functions. And next one is IgM. So the IgM is very important. It is also their own functions are there, that we are called so IgM is very important. It has their own that is functions, that is neutralization. It's also agglutination, is also complement activation. It is also participate in the complement activation. And that is they are depend on the B cell receptor. They are act as a B cell receptor also. So these are the main functions of the M, that is IgM. And next one is IgA. So in the IgA, it is also participate in the neutralization and trapping the pathogens. The pathogens are trapped by the that is IgA. So alpha is very important for the pathogens. They are trapping the pathogens and kill the antigens. So this is the IgA function. And IG so IgE and IgD. So this is the IgD and IgE. IgD is not their main functions are there. We don't know what is the important functions are present by the IgD. So there is no evidence for the function of the IgD, but Ig is very important. So Ig is that so it is very important in the activation of basophils. The activation of basophils is very important, and it also activate the mast cells, basophils and mast cells. It is activate the basophils. So activate the basophils and mast cells, and they are fight with against parasites and allergens. So allergies and parasites, the main is that is antibody E is against their activities are there. Hello students, in this video we are observing that, so this is the antibody. Now we are discussed on the structure of antibody. So the structure of antibody is very important, which is fight against the antigens. So in the structure of antibody, actually these antibodies are released from the B cells. So it is released from the B cells. The B cells are released the antibodies. They produce and release the antibodies. So, but T cells are there, T lymphocytes also there, but these T lymphocytes not released antibodies, they motivate the B cells. So after the entry of the antigens, the B cells are produce the antibodies. So this type of structures, so this is called as a antibody structure. So in the antibody structure, we observe that it look like a Y-shaped structure. So it is a just Y-shaped structure actually. So in this structure, there are two types of chains are there that we are called a light chain. This green type of chain we are called light chain, and the red type chain we are called as a heavy chain. So light and heavy chains means in the structure of the antibody, the two types of chains are present. These are arranged as a Y-shaped structure. Actually antibody we are also called as a immunoglobulin. So that is we are also called as a immunoglobulin. So Ig means immunoglobulin. So in the light chain and in the heavy chain, you are observed that there is a domains are present in the light and heavy chains. The round shape structures, the half round shape structures means half moon like that. Domains. So these are the domains, and these are the domains are present in the light chain and heavy chain. So it is a very important structure in the that is antibodies, and these structures in the light chain. So in the light chain we are observed that that is here the V is there, and this domain C is there. So this is also C and this is also V. So what is the meaning of V? Means variable domains. So the V stand for variable and C stand for constant domains. So in the light chains there is a two domains are present at the either sides that we are called variable domain and constant domain. If present in the light chain, that we are called VL. VL means variable light chain, and VC or CL that we are called CL. VL and CL. CL means constant domain and VL means variable domain. So these are the two domains are present in light chains of the either sides, and in the heavy chain. In the heavy chains we are also observe this type of structures that we are called constant domains and variable domains. So see here in the heavy chain, so here also that is variable domains are present, but you are observed that there are three constant domains are present, that is CH1 that we are called as CH2 and CH3. Now here also there CH1 and CH2 and CH3. So variable region is only one domain present in the either sides, but the constant regions are present is 1, 2 and 3 depending on the antibody types. So in the variable regions, maybe in the heavy chain and maybe in the light chain, you observe that in the that is variable region, that is 110 amino acids are present. So in the heavy chain also contain 110 amino acids, and the constant region also contain 110 amino acids. Means in the light chain the variable and constant domains contain 110 amino acids, but in the heavy chains, but in the heavy chains variable region contain 110 amino acids, but the three domains contain 332 440 amino acids, that is CH1, CH2, CH3. They contain 332 440 amino acids as a double or triple times of the variable regions. So this is the variable and constant regions in light chain and heavy chains, right? And in the structure of the antibody, that is there are terminals are present in the light and heavy chains. That terminals we are called as a here N terminal and C terminal, here also C terminal, and this we are called as a N terminals in the that is heavy chain also present. So these are the N terminals, and these are the C terminals in light chain, but these are the C terminals in heavy chain. So N terminals, and very important that the N terminals we are also called as a FAB pods. So means antigen binding pods. So this we are called. So in the N terminal, suppose N terminal is there. So this is N terminal. Suppose. So here N terminal and here N terminal. So this site and this side of the site we are called as a antigen binding site, antigen binding sites. So in the every antibody it contains two sites, this one N terminal and this one N terminal. There is a two sites are present here and here. So this one N terminal that is antibody binding site, and this one second antibody binding site. Means every antibody binding with two antigens only with the help of N terminals, that we are called FAB pot, that we are called FAB portion or FAB pot. So this is the FAB pot. So this FAB we are called as a fragment anti-binding pot, fragment antigen binding pots we are called as a antigen binding pots. And again you are observed that in the structure of the antibody, this pot we are called as a FC pot. FC pot means this is not binding antigen. It is very important to attach on the antigens membrane. The FC pot is very important to attach antibody on the membrane of the antigens. So this is the second part of the antibody. And in the structure of the antibody you are observe the light chain and heavy chain is bind with some bonds. Some bonds is there that we are called suppose here yellow bonds are there, here yellow bonds are present. And in the domains also, the domains variable domain and constant domains here and here, the yellow pods are as a bond structures are there. So this bond we are called as a disulfide bonds, that we are called as a disulfide bonds. So these sulfide bonds are present here and here and here, and also variables and constant regions. So this we are called as a disulfide bonds. So mainly the light chain is bind with the disulfide bond on the heavy chain, and you are observed that here the two heavy chains are bound with disulfide bonds, or we are called as a interchain bonds. This is called as a interchain bonds. This we are called intra-chain bonds because the light chain is present and heavy chain is present. Between the light and heavy chain, the intra bonds are present, but between the heavy chains, that is interchain bonds are present. So this type of bonds are present. So this region we are called as a hinge region, that we are called as a hinge region. So this hinge region is every time stable. The heavy chains and the intra bonds or intra chain bonds remain the light chains and heavy chains. So this is the structure of antibody, or we are also called as a antibody, or called as a immunoglobulin. So this model is given by, actually this model is given by Porter and Eldman. So these are the very important, that is the structure of the antibody is given by the Porter and Eldman. So these are the scientists get Nobel Prize to give the structure of the antibody.